Now that we have learned how human aging studies are conducted, what types of experimental systems and setups researchers are using, and what type of information they're gaining from these systems, we can look at the actual human aging phenotypes. As is apparent from the previous section, many of the phenotypes associated with human aging, such as muscle loss, cancer, or dementia, very much resemble disease states. Since these diseases have much higher incidence in the elderly, they are often classified as diseases of aging, or alternatively, as age-related diseases. Many geriatric phenotypes, while clearly apparent, do not quite rise to the level of a disease state. For example, we know that certain plaques form in the brain during aging, but brain function remains unimpaired. In other individuals, these plaques lead to functional loss and Alzheimer's disease, which begs the question: what if aging is the disease and Alzheimer's disease, cancer, etc., are merely the symptoms? According to Merriam-Webster, a disease is a condition of the living animal or plant body or one of its parts that impairs normal functioning and is typically manifested by distinguishing signs and symptoms. But that is a dictionary and not a scientific definition. From a scientific or medical point of view, disease is quite difficult to define. In fact, the WHO, the World Health Organization, has a definition of health as a state of complete physical, mental, and social well-being, not merely the absence of disease or infirmity. So more and more, scientists view aging as a disease of its own, a disease that may be treatable just like any other disease. In fact, in terms of R&D investment, it may be more cost-effective to develop interventions that treat aging itself, rather than finding treatments for every single age-related disease. Rather than treating every geriatric disorder, symptoms, and disease on its own, we could treat them all at once if we address the underlying ultimate cause of disease, which is the aging process itself. The goal of such research would not be to increase longevity and lifespan per se, but rather increase what is called health span, which is the period of time an individual is in good health. In fact, it is counterproductive to increase someone's lifespan to 200 years if that person is frail for the last 100 years of their life. It certainly is preferable to live shorter lives, but with much reduced frailty period at the end. The idea is to add life to years and not years to life. As mentioned earlier, we will talk about anti-aging interventions in the final module of this course. In later modules, we will examine in more detail some of the organs and tissues most affected by aging. But for now, let's look at some examples of age-related diseases. One of the most important diseases of aging is arteriosclerosis, the hardening of the blood vessels. The cardiovascular system is responsible for transporting blood from the lungs, where it is loaded with oxygen, to the tissue, where that oxygen is used in mitochondrial respiration for energy production. It consists of blood vessels of varying diameter, from the very wide aorta near the heart to the capillaries in the tissues, which are just wide enough to accommodate just a single red blood cell. These blood vessels are surrounded by a layer of smooth muscle cells that allow the adjustment of vessel diameter according to the physiologic needs of the organism. During aging, the ability to constrict and expand diminishes, partially due to an increased level of cross-linking of collagen fibers, the primary proteins of the skin and connective tissue. Highly cross-linked collagen loses elasticity and makes it more difficult for blood vessels to adjust their diameter. So, cross-linked collagen leads directly to blood vessel aging and, in fact, is also a major culprit in skin aging, making skin less elastic with age. The most important form of blood vessel hardening is arteriosclerosis, the buildup of insoluble plaques inside blood vessels that restrict blood flow through the vessel. When blood flow through vessels near the heart is restricted, ischemia is the result, which may lead to heart attacks. When blood vessels supplying blood to the brain are affected, stroke may be the outcome. These events are a major cause of death in aging populations, especially in developed countries. There are important environmental components to this disease, such as diets high in saturated fats and cholesterol, smoking, or high blood pressure. However, there are also important genetic risk factors involved in the development of atherosclerosis. For example, the ability of cells to self-renew declines with age. And this is something we will talk about in more detail when we discuss stem cells and senescence. Thus, cells that normally clear up plaques become less abundant. Epithelial cells lining the blood vessels may not be replaced with newer ones. Or even some immune cells may get locked into a pro-inflammatory state that damages those blood vessels. While the exact mechanisms are still unclear, there's evidence that all these scenarios contribute to vessel hardening with aging. One such genetic biomarker for atherosclerosis, but not only for that, but also for aging, is called ApoE, or apolipoprotein E, one of the proteins that is responsible for carrying lipids, fat, and cholesterol through the bloodstream. Another interesting phenotype of aging is muscle loss, or sarcopenia. Similar to other aging-related phenotypes, it is multifactorial, containing both environmental factors, such as a sedentary lifestyle, as well as genetic factors. Interestingly, one of the genetic factors associated with muscle loss are mitochondrial genes, and those affect mitochondrial function and efficiency. Mitochondria are the power plants of the cell, creating the vast majority of cellular energy in the form of ATP. If mitochondrial function is compromised in muscle cells, the muscle will not have sufficient energy to function and thus will begin to atrophy. As a consequence of this loss of muscle mass, less energy is converted into muscle movement, and surplus energy can now be stored as fat instead, which in turn creates another aging-associated phenotype, age-related obesity. This pathway can also occur in the reverse, where obesity in turn leads to muscle loss. Related to skeletal muscle loss is bone mass loss and osteoporosis. Adult bone is continuously remodeled via the interplay of osteoblasts, cells that make new bone tissue, and osteoclasts, cells that break down bone tissue. Since these are cell-based processes, it is highly likely that they have their origin in deficient bone stem cells that are less capable of creating new osteoblasts. Both genetic and environmental factors may contribute to this disease pathology, especially dietary calcium intake, which is critical for the formation of the bone mineral hydroxyapatite, and also exercise, or rather, the lack thereof. Interestingly, collagen cross-linking, just like we talked about earlier for vessel and skin aging, may also play a role in bone aging, because collagen is an important component of the bone connective tissue as well. Finally, as a last example of aging pathologies, COPD, or chronic obstructive pulmonary disease, serves as an excellent example for the pleiotropic nature of the aging process. COPD is a chronic inflammation of the airways, often caused by environmental factors, especially smoking or air pollution. From an aging point of view, several mechanisms are at play here that contribute to disease pathology. COPD is characterized by chronic inflammation that is an overactivation of components of the immune system. While the immune system is clearly needed to remove pollutants from the lung, these very same processes also damage the lung. In addition, pro-inflammatory cells often release radicals, chemicals meant to fight off foreign invaders. Once again, too much of a good thing turns out to be a bad thing and causes tissue damage. Finally, this inflammation leads to increased proteolysis, the breakdown of proteins. If cellular proteins are broken down, and not the proteins of the foreign pathogen, protein homeostasis will be disturbed, and once again, that leads to tissue damage and tissue dysfunction. What these few examples clearly demonstrate is that age-related diseases are very pleiotropic in phenotype and highly complex and multifactorial in cause, with both environmental and genetic factors contributing to disease pathology. For example, collagen cross-linking increases over age, as certain toxic byproducts of metabolism are not efficiently detoxified. The reason for this failure of detoxification, in turn, can be multifactorial, and we will explore those mechanisms in Section 3. But since collagen is such an important component of so many connective tissues, including skin, bone, and the vascular tissue, any tissue that contains collagen will be affected by molecular collagen aging. Similarly with muscle loss. Loss of muscle mass leads to reduced grip strength, generally poorer performance on strength, speed, or endurance tasks, but it also affects vision, as the function of the muscles that adjust the thickness of the eye lens is diminished. In turn, vision impairment has multiple other causes, from macular degeneration to cataracts that damage the lens. However, certain common themes emerge here. For example, in COPD, inflammatory immune cells remain hyperactivated and damage lung tissue. In many other aging-related disorders, pro-inflammatory cells are observed, and thus aging is often viewed as a pro-inflammatory state, often called inflammaging. In addition, depletion of stem cells, such as we've seen in bone aging, is another recurring theme. In fact, the loss of these stem cells, the role of which is to proliferate and regenerate many tissues, is another common aspect of many diseases of aging and of the aging process itself. On the flip side, dysregulation of proliferation ultimately leads to cancer, another aging-related disease. Finally, loss of homeostasis, in other words, the precise balance of cellular processes, be it metabolic, protein or DNA homeostasis, leads to abnormal cellular function, which in turn affects then organismal performance. The question remains, what is the ultimate cause? And what are the molecular pathways? As we explore these questions in the following modules, you will learn about the molecular events underlying these disease pathologies. You will also see molecular examples of all these evolutionary theories of aging. In the upcoming section, we will focus on the big three: how stress affects aging phenotypes and what stress is on a molecular level, brain aging and Alzheimer's disease, and finally, endocrine aging and type 2 diabetes. Check your understanding. Explain the difference between aging and disease. Explain how changes in a single cell/single protein can affect aging phenotypes in multiple organ systems. Describe aging phenotypes of common human age-related diseases.